Shiny, shiny, shiny. We are here so shiny. We are shiny, shiny science, shiny people, shiny happy. I'm done. Okay, this is the TWIS podcast.
You do know we're live. Yeah.
Oh, what?
Yes.
We're what? With who? For how much? Hi, everybody. We're here to do the TWIS podcast. So I think we'll start that now. It's live with ads. So I'll give you Twitchers and others who are potentially watching on ad-generated platforms. I'll just say, look at my hair is going frizzy. I'm just giving you a moment before I actually start the show.
Are we stalling for them to... Can't they by now? They've got to be done with whatever ads exist.
They're done with whatever ads were in existence.
Goodness. Thank you so much for putting up with our kind sponsors. How do you say it? I don't know the... right.
Pre-rolls.
I have the wrong tact for
30 minutes. Don't worry about it.
Apologizing.
But if people are here right now, they're here right now to see This Week in Science on our live broadcast of the podcast that we do every week on Wednesdays at 8 p.m. or maybe starting a couple of minutes later plus 30 seconds for pre-roll ads if that happens on the platform in which you are watching. Now, we are going to start the podcast, which is not the podcast, but the broadcast of the podcast. And the podcast is going to be edited from the broadcast, which we're going to do right now, starting in 3, 2, This is TWIS. This Week in Science, episode number 830, recorded on Wednesday, June 23rd, 2021. Will the aliens find us first? Hey everyone, I am Dr. Kiki, and tonight on the show we will fill your heads with butterflies, birds, and mice. But first...
Disclaimer. Infrastructure. The brick and mortar backbone upon which the modern world depends to carry the weight of our fast evolving civilization. Constructed from the ingenuity of human engineering and design. That functional visual manifestation of Earth's one sentient being. Reimagining the surface of the one habitable planet in our solar system. And, as it turns out, our infrastructure is crumbling. Not entirely crumbling, not all at once. In many places, the infrastructure might actually be brand new. But everything we build has a shelf life. A number of years before nature reclaims the material we build with and returns them to the earth. Every crack in a foundation, every dusting of rust on an exposed bit of rebar, along every mile of road from sea to shining sea, and in every pothole along the way, there are signs that nature remains relentless in its push for entropy. And our best response so far has been cement. Cement is most widely consumed material globally. It is the industry itself accounts for 8% of all human-caused greenhouse gas emissions. So as the politicians decide how best to spend infrastructure dollars, let us not overlook where those dollars could be best spent. Research science. I know you're surprised that that's the answer I came up with, but research science is where we should be putting our dollars. Angie Therese Acano at the McCormick School of Engineering is a research scientist who is working on ways to make cement stronger, longer lasting, and better for the environment. She is an example of what we actually need as an integral part of infrastructure planning. Material research that can lead to lower maintenance costs, lower greenhouse gas emissions, and taller structures. By 2050, the United Nations predicts 2/3 of the world's population will be concentrated in cities. So, either we build up or we pave the rainforest. And roads. We could afford every good thing humanity deserves if we could just spend less maintaining roads. If you don't know, the national average is $25,474 in transportation spending per mile per lane of road. And hey, who knows? With all the money we save by funding research that goes on to save us money, we might be able to spend even more money on research. And the more money we spend on research, the more we will have of This Week in Science coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill
Science to you, Kiki, and Blair's gone again! Man.
Blair is out again for another evening, and we do wish her well.
We do. But just so you know, it's going to be Justin's Animal Corner today. I'm taking it over.
Justin's? Hold on.
Justin's Animal Corner. Justin's Animal Corner with Justin. It's got a nice ring to it. I like it.
We'll see what Justin's Animal Corner gets up to. Probably not as many invertebrate stories as Blair, but we'll see.
I didn't want to make her mad at me. I want to right?
never make the Blair mad.
No.
Oh, okay, everyone. This is This Week in Science. Good science to you all, and thank you for joining us for another week of fun-filled science discussion. Woo! We have all sorts of amazing stories for you this week. I'm going to be talking about alien planets, some quantum standstills, magnetic birds, and mice and brains. And Justin, what did you bring?
I've got retroviral communication. Tiny T-Rex bird brain, or skull, not a brain, just the skull. Ignoring COVID.
At your own peril.
Record-setting butterflies, toxoplasmic hyenas, and why not to shake hands with your veterinarian.
Oh, okay. I will keep that in mind next time I have to go to the veterinarian after you tell me that story in just a little bit. For those of you who are just joining us and haven't done so before, just understand that you can subscribe to This Week in Science on YouTube, on Facebook, on Twitch, and on Instagram and on Twitter. And we are also a podcast on all the places where podcasts are found. So look for This Week in Science. Our website is TWIS.org. All right. Ready for the science, Justin?
Let's bring it.
Okay. Let's start by taking a nice look at the Earth from the perspective of other stars. That's a new thing to do. Researchers have previously tried to figure out how the Earth might look from other planets, from other places in the universe, from the perspective of aliens that might be gazing out at the stars and trying to figure out if they are the only life in this universe. Previously, though, scientists had looked at the stars and been like, nope, those stars, there's only a few that would be able to take a good look at the Earth, and, well, none of them probably have Earth-like planets or life-producing planets on them, so we're all good, no big deal. However, some new researchers decided to cast a different kind of gaze around the universe, and they reported in Nature this week their work that took a look at not just the perspective from the stars, but from the perspective of stars that would be in the plane of our galactic orbit. So where our solar system orbits around the Milky Way and how our planet is orbitally inclined around our sun, how our solar system is inclined, to think like Earth humans gazing out at the stars and using the Kepler space telescope to get ideas of what exoplanets might be transiting distant stars.
Because my initial thought was, well, gosh, isn't it just like if we can see a star, they can see us. It's just solved it. But the way that we find these other planets out there is they have to go kind of like across in front of they have to pass in front of their sun.
Yes.
And that's when we get the little dimming blippity effect. So what we're looking for, it's the stars that we can see when the sun is at our back.
Exactly. So, and it's not just when the sun is at our back, but also when we are in the right plane of viewing for distant stars. So not all stars are going to be looking at us at the right potential time. If there is a star gazing down at us. All these ups and downs are relative because
very difficult in space to say an up and a down.
Difficult,
right? The angle has to be right.
The
too.
angle
has
to be exactly
enough
right.
Angle that we're looking out at.
Yeah. So these researchers have found approximately 2,000 stars. Some 1,715 of them have or are in the right position to have spotted the Earth in the last 5,000 years. In the next 5,000 years, they estimate they've found another 319 stars that might be able to take Earth in as a view. And of those 2,000-ish stars that might be peeking at the Earth within the plus or minus 10,000 years surrounding our here and now, 75 stars within about 30 parsecs have already been potentially swept past by our radio waves. All of our noisy radio and TV emanations that we've been broadcasting to each other have also broadcast out into space, and some 75 stars may have already heard us. We can take that number down a little bit further, and there are around 25 stars that could possibly have a view of us right now and be able to see us. There are some stars. Do you remember the discovery of the TRAPPIST solar system?
That really weird tiny planet with lots of them and really a tight where there could have been multiple a Goldilocks range.
Yeah, multiple Goldilocks planets potentially in that system. And in about 1,600 years, the TRAPPIST-1 system will be able to view Earth for a total of about 2,300 years. So there will be, it's a long time from now, but 2,300 years in which they could put any life in the TRAPPIST system gazing out could potentially view us if we're still around. Now, there's another star.
But even if we are, there's still that little hiccup that we're actually we seem to be getting quieter. We're not broadcasting. We're still doing radio for now, but a lot of radio is actually digital.
Right.
The television is
that blip is gone now. A little bit.
The blip is. We could get to a point where we're not broadcasting anymore.
We might, but we still have radio waves to some degree. But researchers, though, they've said the researchers involved in this have said maybe we should think about putting a transiting megastructure in place for the TRAPPIST system to observe in the year 3663. And that if we put, instead of just our planet Earth, if we have a transiting megastructure that's larger than Earth, it'll really dim the light of the sun and get the attention of any aliens in the TRAPPIST system. Now that's if we want to go down the sci-fi route and make contact. And, we'll see where we are in some 1,600 years as to whether that's something that's interesting to humanity. But it's an interesting new way of considering the universe around us and any potential life that might also be looking into the stars and what they'd be able to see and when. This is what we're doing. We've found exoplanets now. Who else might be doing this too?
It's an intriguing question, I suppose.
It is intriguing. And this gets, and then it dives into deeper questions of, who are we going to choose to make contact? Are we going to choose to hide? What are we going to do? And there are many people with many different opinions on this Earth. So it's a big question for humanity as we reach out into the stars. But as we're reaching out, Justin, I think your next story had something to do with the reaching in, maybe little tiny things inside of us reaching out to talk to us.
So, this is a pretty interesting story. National Institutes of Health scientists and their collaborators have identified an internal communication network in mammals that may regulate tissue repair and inflammation, providing new insights on how researchers can tackle diseases such as obesity, inflammatory skin disorders, these sorts of things. This research is published in Cell. So there are billions of organisms living on body surfaces and the skin of animals. Collectively, we refer to this as our microbiota. They communicate with each other and their host immune system in a sophisticated network. According to the study, viruses integrated into the host genome. These are remnants of previous infection. This is what we were talking about when we were talking about endogenous retroviruses. These virus remnants in our DNA can control how host immune system and microbiota interact and ultimately affect the response to tissue repair and antimicrobial defenses. So, retroviruses in your DNA aren't a casual component. They can comprise as much as 10% of all of your genes. The newly discovered role of these retroviruses adds to a sort of long-standing, or a building. I guess it's not really that long-standing. I guess since we've started this show, it feels like it's been going on quite a long time. But it's been basically during the life of this show that this type of research has even come to light. So they say here, this is from the authors. Together, our results support the idea that mammals may have co-opted their endogenous viromes as a means to communicate with their microbiota, resulting in a multi-kingdom dialogue that controls both immunity and inflammation. This is not a flyby study by any means. These are scientists from NIH's National Institute of Allergy and Infectious Diseases. They led the project. They had collaborators from the National Institute of Health Center for Human Immunology, the National Cancer Institute, Stanford University, Scripps Research in California, University of Pennsylvania, University of Oxford, the Francis Crick Institute in England were all part of the collaborators on this project and this research. Yeah.
Huge.
Yeah, so building on the series of studies over the past decades showing that the microbiota broadly promote immune protection, this is, and as well as some human behavior aspects to it and everything. Like we've been talking, if you've been listening to this show for a while, you've heard us going on and on about the microbiota. The scientists and collaborators tried to do that thing that scientists do. They asked the question, why? They looked for the mechanism. Using Staphylococcus epidermidis, a common skin bacterium with known helpful and harmful features, as a study model in laboratory and mouse experiments. The models helped them identify the role of skin cells, keratinocytes, and the endogenous retroviruses in communication between microbiota and skin immune system. This is just like, I mean, we know that something's going on, so it shouldn't be like, wow, this communication is taking place through this mechanism that we wouldn't have imagined. But of course, that's always sort of the case. Keratinocytes are the primary interface between the host and the microbiota. Their studies showed that epidermidis triggered an antiviral response in the keratinocytes, and that finding led them to discover that the retroviruses coordinate responses to the microbiota that stimulate then the immune system. Mouse model also showed that a high-fat diet triggers an inflammatory immune response to the S. epidermidis that can be controlled by providing an antiretroviral treatment.
Huh.
Yeah, so that means there's a way to block the communication of the retrovirus from driving an inflammatory response caused by microbes who are under high-fat conditions that would then be... But that is just absolutely mind-boggling. I mean, I don't know how... I couldn't even give you a guesstimate on the number of diseases that are inflammatory response diseases.
I just feel like there are so many, and diseases that we didn't think were inflammatory diseases are coming to light that they are inflammatory diseases. So it's
or has an inflammatory aspect.
Yeah.
Yes.
Yeah.
It's somewhere. It's like there's a retrovirus in the DNA from the virus. It's like, yeah, we've got something for that. I don't know if you wanted it. Well, you know what? Just send it. I'm just going to go send an inflammatory response.
Yeah. Yeah, it's really amazing, though. I mean, it does make sense that we would have these endogenous systems where our body would be communicating with the microbiome, with the microbes that are on and in us to coexist. That our body and those microbes have been evolving together for a very long time, and these systems have, For the longest
time, the retroviruses in our DNA we thought was just, oops, oh well, it's there. It doesn't do anything. Obviously, we're not producing virus off of it.
But it could be helping us deal with microbes.
Yeah.
Yes.
And so then there's that whole thing where microbes can swap, some genetic DNA that they incorporate. And of course, that seems like, well, if you're an amoeba or something, you might be able to do that. But humans, we're so evolved. No, we've been doing this to get this evolved. This has been part of that process.
We've been swapping genes so that we could get to where we are. And everybody's swapping DNA. That's the way it goes these days. Except for those moments when everything is at a complete standstill. But I mean, really, when you think about that, does that ever happen? No. I mean, even when you look at the tiniest microscopic aspects of the universe, go down to that quantum level, it's vibrating. Everything's moving, constantly moving. But researchers have been trying to, over time, bring matter to a standstill. And we have supercooled various atoms, things at the atomic scale. We've been able to use magnets and all sorts of energy, really, to reduce that movement. And so when we think about what temperature is a measurement of movement. It's it's a measure of energy in a system. And temperature is really like this difference, this differential between things. But researchers have been able to get atoms down to super small scale, super small nanokelvin levels. And when they get down there to this point of almost not moving anymore, this is called the motional ground state. Now, researchers at MIT and other organizations have used LIGO, the Laser Interferometer Gravitational Wave Observatory, not to observe gravitational waves, but to create a functional, cooled large object. Basically taking something the size of LIGO and its mirrors, which we have this laser interferometer, laser light bounces off these very, very, very sensitive mirrors. And in bouncing off those very, very sensitive mirrors, these tiny little bounces of the laser give us this idea of how much our planet is bouncing based on due to gravitational waves. They have been able to take this interferometer and control it so that they know when one photon is bouncing off a mirror and how that affects another photon's movement. And in understanding the movements of the photons bouncing off the mirrors, they're able to, with motors, stop the vibration of the mirror and, in effect, stop the vibration of those photons. Like as if like a car accident, or if there's a ball that's being thrown at you and you suddenly stop it with your hand and it just stops there, that moment before it bounces away. That stoppage of movement and taking all of photons of light and taking all 4 mirrors of LIGO, which are like 40 kilograms or something in weight, the average comes out to an object. This is a virtual object because it's taking all of these 4 mirrors from LIGO and putting them together, that would weigh in at about 10 kilograms, which is the size of something reasonable. Like, that's a decent size object as opposed to something at an atomic scale. And in stopping the movement of these mirrors, they have fundamentally created emotional ground state for an object that is virtually the size of a large piece of matter, a 10 kilogram piece of matter. And now having this stoppage of motion created an effective reduction in this virtual object's temperature down to about 77 nanokelvins, which is like, this is unheard of. And in doing this, now they think that, why are they doing this? What have they done? This is weird. What have they done? This is just a random big experiment. And okay, you can stop the mirrors moving and the light bouncing. And then what happens? In doing that, what we will be able to do is test in the future the effects of gravity on quantum objects that are large sized, like the size of your desk, like the size of a car, like the size of a laptop, as opposed to atoms. So it's going from the atomic scale to real world scale, which is kind of exciting for physics. Yeah. I'm excited.
Somebody in the chat room is not familiar with kilograms. Yeah, 10 kilograms is approximately 10,000 grams. So it's about the same.
Thank you for that clarification. I appreciate that, Justin. Yeah. Yeah. So it's a big story. Using LIGO for something other than gravitational waves is a unique adaptation of the technology, and I don't know. I'm excited to see where they take it. Tell me about, though, we're not going to talk about quantum stuff anymore. Tell me about birds. Do you have a dinosaur or a bird? I'm confused.
Well, that's the question. I don't know. Researchers from the Institute of Vertebrate Paleontology and Paleoanthropology of the Chinese Academy of Sciences have discovered a 120 million year old partial fossil skeleton of a tiny extinct bird. This is very tiny, so small it could fit in the palm of your hand. And it has the skull of a T-Rex. Well, at least it has a unique skull with a mix of dinosaur and bird features. Those dinosaur features being functional features that are shared with Tyrannosaurus Rex, kind of showing that this early bird kept many of the features of the predecessor. So their findings are published in Nature Communications, June 23rd. The bird was deposited 120 million years ago in a shallow lake in what is today Liaoning province in northeastern China. Through a detailed reconstruction of the bird family tree, the researchers demonstrated that the new fossil bird species belongs to an extinct group of birds called Enantiornithines, or opposite birds. They are the most diverse group of birds from the time of the dinosaurs in the Cretaceous and have been found all over the world in some form. In living birds, the quadrate is one of the most movable bones in the skull. It allows for the unique feature of living birds known as kinetic skull, which allows the upper jaw to move independently of the brain and lower jaw. In contrast to living birds, the skull of the new opposite bird was as well as those dinosaurs like T-Rex and the close dinosaurian relatives of birds, they were not kinetic. Instead, the bones are locked up and unable to move. So the temporal regions, sides of the skull of this bird fossil, very different from living birds. Species has two bony arches for jaw muscle attachments, like those found in reptiles. This is lizards, alligators, dinosaurs, making the rear of the skull rigid and resistant to movement along those bones. I don't see here. The one thing I wanted to know, which I don't see here, is if it had teeth.
And this wasn't just a little tiny baby? This wasn't just a little teeny tiny baby T-Rex?
So no, it's not. But you know what? That's a great point you bring up, because one of the things that has foiled paleontologists in the past is the reliance on morphology. If a thing looks a certain way, it's a certain thing. And one of the things that they discovered, especially common amongst dinosaurs, which is still something you can see amongst mostly in certain bird species today, is the ability to both grow and then absorb and redistribute bone material. Something that we all humans do, all mammals do all the time. You wear your bone and it grows a little bit more. If you haven't used a muscle, the bone that attaches there might atrophy a little bit. That material or that energy to produce that calcium goes somewhere else in the body. But that through normal life stages, they have found dinosaurs that would have these big cranial ridges that then would disappear and might show up in the back of the skull somewhere, or might just be gone completely, or they would. So morphologically, I agree. This might be a little baby bird. It might be a fully grown bird. It might be a species that they've found before that looks new because of it's happened enough times. I think there's some classic examples, but Triceratops, like that
right.
Like we had like a dozen or more of them. That it turned out, well, sometimes we're looking at adolescents, sometimes we're looking at adults, sometimes we're looking, but the morphology of the horns and everything actually could change so drastically over the different ages that you weren't always seeing what you thought you were seeing.
Do you see what I see? I see a dinosaur-skulled tiny bird.
Oh, there's the little teeth.
And it's got little teeth. It does. Little tiny dinosaur teeth.
I didn't see anything. It's in the. You found a nice picture of it. I was what I was reading. I didn't see it specifically talking about the teeth, but the skull and jaw muscle attachments made it sound like a biter.
So this, and so if it's a biter, if it's got the large muscle attachments, even though it's a tiny skull compared to what a T-Rex skull would be, a miniaturization, it's still, if it's got the large muscle attachments, you would expect to see teeth or something that would be, this is biting, and it's going to, this little tiny bird would be fierce. Super fierce little animal. I love it. That's really fun. I have another bird story about magnets in birds. It's in their eyes. We've talked lots of times about how birds migrate and how that migration is very possibly made possible by a special sensory power, the ability to be able to sense the Earth's magnetic fields. And it's been a big question as to just how that is possible. How on earth do these migratory birds potentially see the magnetic fields? How does it work? Well, a while back, we also reported on a study in which researchers looked at proteins, cryptochromes, which are light-sensitive proteins that are in various eyes. Human eyes was where this study went, and I think it was last year. I don't know, it was a few months ago, but the study found that humans have cryptochromes that when they are exposed to light and magnetic fields, they change formation and thus are sensitive to magnetic fields. And so everybody's like, why do people have cryptochromes that are sensitive to magnets in their eyes? That's weird. And then this led to these researchers taking cryptochrome 4 from the eyes of migratory European robins and taking a look to see whether or not they were sensitive to magnetic influence. They compared this cryptochrome 4 against 2 non-migratory bird species, one chicken and the pigeon. Very non-migratory, although pigeons have a good homing sense for homing pigeons. They don't migrate. And so why would they need magnetic fields? Chickens, they can jump into and out of trees very nicely. They don't need magnetic fields. So they tested the mutations that were in these different cryptochromes and tested how the cryptochrome formation changed when they were in the presence of magnetic fields. And they discovered that, yes, indeed, the migratory robins have a magnetic field sensitive cryptochrome. So we don't know exactly what that would look like in their visual field. It's been talked about as it like maybe it would appear as like a glow or some kind of, what would that be? We can only imagine, right? How that would lay over.
I immediately pictured it almost like you're saying like a glow, but certainly like if you had a GPS that just pointed you in the right direction. Like,
exactly.
Nope, that's not it. Oh,
this is the way you go. Go over there.
Oh, there it is. I can see it now. Yep, this is the right way. That's when it gets all shiny.
Yeah. So they're going to do more studies and try and figure out exactly what's going on. But this really is starting to point to, and this is weird because we've been talking about this for so long, and to see it go from iron particles in the beak of the bird to suddenly these light sensitive pigments in the eye and these pigments are sensitive to magnetic fields. This is just, it is wild. And I mean, probably decades ago, researchers never would have considered this possibility, but this could potentially be a mechanism for how the magnetic sense works in birds.
So, and while we're talking about a heads-up display or whatever it is, the other thing to always remember is that the thing that's occurring is that the eyes are just a sensor. The brain does all the work. Like it oddly reminds me of this long history. It started with this. There was a shaman who could repair vision. He wrote books about how to fix your vision because you have to look like an eagle, move your eyes rapidly around, and then fixate on something. And Aldous Huxley. This is a long story. Aldous Huxley was going blind as a child, read on this, did the exercises, and it worked. But he's like, I don't believe in this eagle eye shamanism. It's because I've strengthened the muscles. Right. And then years later, they did research on people playing video games, see how horrible it is for your eyes. Turns out it was great for your eyes if you were playing one of these games that had lots of motion in them. But it wasn't repair on eyes. It's the fact that the brain got better at processing the information. And there's so much that the brain is doing involved in vision that we're not the least bit tuned to or aware of.
It just happens.
Most
of our vision does not happen in our eyes. It happens in our brain.
It's all happening in your brain. Yeah. This is just this is a port that the light gets information starts to come into, and then it gets translated. So that long story short, that heads-up display that's taking place in these birds may not be visual at all. It could just become sort of like an impulse or an instinct or a feeling. Could be just a sense.
Yeah.
Because the brain doesn't need you to see everything it's learning or processing or doing. It doesn't need to communicate back, I guess, to you with a visual stimuli.
You don't need a heads-up display.
You don't need a heads-up display. You have
when it's integrated. When it's
that integrated.
Yeah.
Don't need to show it back to you. You are just there to gather the data. The brain now can do all of the rest of that work without you knowing it.
This is This Week in Science. Thank you, everyone, for watching, whether you're on Facebook or YouTube or Twitch. If you're watching live, we appreciate you watching right now. Thanks for being a part of the show. And if you can, and you like the show, tell a friend. That'd be great. All right, it's time for the quick COVID update. I don't think that music goes with the COVID update.
I think it's lovely.
At all.
I think that's the best type of music for a COVID update. Thank you.
Ah, you're welcome. Okay, so on Twitter today, I became aware of a New York Times story that was based on research that just was published in the bioarchive based on a based on the work of a molecular evolution laboratory, the J. Bloom Laboratory, that's affiliated with the Fred Hutch Research Center and HHMI. Now, the Bloom Lab study published their study in bioarchive, recovery of deleted deep sequencing data sheds more light on the early Wuhan SARS-CoV-2 epidemic. And now you might see people making a big deal out of this, saying, oh, this points more to the lab leak, and trying to cultivate more controversy and more more trouble. Now, number one, it looks as though there is an NIH-maintained database called the Sequence Read Archive. Scientists can put their deep sequencing data in this archive for other scientists to peruse. The Bloom Lab went about checking through the data from last year related to a paper that was published on the PeerJ open access journal, and in doing that discovered that there was data from a project by Wuhan University that had been deleted from this NIH archive.
Dun, 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 dun.
It's not a terrible deal that it was deleted. Researchers delete data all the time for various reasons, and there's a process that you can go through to email the archive and say, hey, I kind of messed up on that or whatever. Can you delete that? And it'll get deleted. So we don't know why the data was deleted, but data was deleted. Second, the data is...
Say it right back.
The data actually does not support a lab leak necessarily. What it does support is this came from bats, and that the sequences that had been deleted were actually more closely related to SARS coronaviruses that have been found in bats than the sequences that had been taken from the Wuhan market. And so researchers are now saying, all right, so the whole idea that it came from the Wuhan market, we're putting that to bed. And they are starting to look at the bigger idea of there's a lot of bat-based coronaviruses around, and it's very likely it could have come from other areas, come into Wuhan, and that's when we first really became aware of it. And so there are questions definitely as to whether or not China was deleting research that it hadn't gone through. The Chinese government likes to have a very specific publication process where everything is vetted and okayed by the government before it gets published, especially for something like the pandemic. And they wanted to cover their tracks in case it was something they did wrong, maybe. So it's very possible. I'm speculating here. But definitely some things were deleted. We don't know why.
So
we don't know. But it does not. This is not this is the evidence that it's a lab leak. No, don't even listen to people who are saying that. Go ahead, Justin.
Could be a lab leak, but
most likely not.
Well, but the thing is, like, the Chinese government does have a bad track record, and I don't think it has to do with about if they did something wrong. I don't think that they care. It has to do with business, keeping business and tourism going and all these sorts of things, and they don't want to slow that down. So their don't talk about it policy is not great, and you could point to that and say, aha, that's why you can't trust it. But then you look at, like, how many governors and the president of the United States was like, let's not test, and then that means that we don't have a...
There's all sorts of political... Yes,
politicians with good public health decisions have a bad track record generally. One of the things, though, when you're talking about trying to get to ground zero on a pandemic is that pandemic, not even before it's a pandemic, but when it's just a spread of a disease like this that's transferred from the animal world into the human world, is that they don't make that leap and then just run with it. They make that leap and it fails, and they make the leap and it fails, and they make the leap and it fails, and they make the leap, and then there's a little bit, and then it goes away because those people don't transmit enough. And so there's such a staggered effect that when you're talking about a city with the population of 2.5 million more people than New York City as a ground zero, I don't think we appreciate when the beginning was. I think this idea that we have this beginning when the virus is in people and spreads easily amongst people belies how viruses transmit once they've made that leap. It's very likely that we would need to go back, look much further back, and probably, because of the interactions of a city that large, you would also need to look at what population of people had an interaction with that city. And what you'll find is pretty much everywhere.
Everywhere. Everyone. Yeah, it comes from all over.
With that city. So the idea that you are going to trace it back, the only way to do it, and I'll just wrap that up. The only way to do it is if you find the natural reservoir, because then you have a potential idea of a starting place. And
if you find the reservoir, then that's your smoking gun, right? You're like, there's the reservoir. There's the genetic sequence we're coming from. That's exactly it. We don't have that yet. We just have loose connections at this point. Yeah.
And until you have the reservoir, you can't then even know where you're trying to trace back to interactions to. So it's like an impossible problem. There's too many puzzle pieces, and they don't all belong in the puzzle. That's the problem.
Yeah. And like our guests from last week mentioned, there is a paper published in Cell this last, actually, I don't know if it's actually published. The embargo, it does not have an embargo, so it's available. Identification of novel bat coronaviruses sheds light on the evolutionary origins of SARS-CoV-2 and related viruses. The researchers studied 411 bat samples that were collected in Yunnan province in China between 2019 and 2020. They got 24 full-length coronavirus genomes that had 4 viruses that were very, 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 very related to SARS-CoV-2. 3 of them were very related to SARS. And the closest relatives to SARS-CoV-2 infects a species of bat that is found in regions extending from South Laos and Vietnam to southern China. So this is the study actually starts getting at, hey, look, here are populations of bats that overlap this area that have related viral sequences. And so there are bats that we should be monitoring, and maybe this is the direction that we need to be going to find that reservoir that you're talking about. But the work is happening. But so there are sticking points because of politics and politicians having different ideas about the way things should be done than the scientists. And that is an issue. But yeah, hopefully the scientists will come out on top.
Yeah, I could hope. The problem is science never flexes its muscles. Science can have its funding cut off. Scientists can be silenced by authoritarian governments. Science never goes, you like electric light? You want electricity?
Once science has made the discovery,
your asthma medicine? You like your allergy pills?
Yes.
You like those? You wouldn't want anything to happen. Say, oh gosh, suddenly you don't have your allergy medication. Can't go outside anymore. Oh, and it sucks because you got to be inside and no lights. What are you gonna do? Like science never. Sorry, it was fun.
And then, but moving on from the source and understanding that our understanding is only as good as our data happens to be. We've got researchers now who are working with the National Institute of Health to study people long terms here in the United States who have who may have previously been infected with COVID-19 but didn't know it. So they looked at a large sample of people representative of the general demographics of the United States, and they found that there were a massive number of undiagnosed cases. They discovered this by looking at antibodies in people's blood. They got people to submit blood samples, and they discovered, wow, like 16 million more people had COVID-19 by last July than we had thought, which is a significantly higher number. And if that significantly higher number is transferable to the rest of the pandemic because of asymptomatic cases or cases with very low amounts of symptoms, this is potentially going to give us a different outcome with the variants that are coming through now, the variants of concern like the Delta variant that we're worried about. And researchers are going to be following up with these participants to find out more about how vaccination affects future infection rates, and also how naturally acquired antibodies affect responses to these variants of concern. But anyway, yeah, higher seropositivity, which was thought to have happened, but it wasn't exactly sure. And then Francis Collins, the head of the NIH, just summarized a recent study that compared the antibodies that were produced by vaccinated individuals versus those people who had been infected but not vaccinated.
Oh, interesting.
And they discovered that, and so we've had this question on the show for a while of like, all right,
how come we don't know? Why don't we know that there's enough people infected? You should know if they have the antibodies, if they have the immunity, versus like if they were just vaccinated.
But then there's also the question of if you're naturally acquiring, naturally means you're getting infected, right? Versus getting vaccinated. How is that going to influence your immunity? Is one better than the other? And there's a big question about that. And what they've discovered from studying the antibodies in the blood of these people, like a really in-depth analysis of what antibodies are in these people's blood, they found that there actually is a striking difference in the types of antibodies, what the antibodies have been constructed to respond to in the blood of the naturally acquired versus vaccine acquired antibodies. And there's a part of the spike protein that is the binding domain. And the binding domain is really important because it latches on to the ACE2 receptor.
You're killing me.
What?
You're killing me. Which one's better?
They think that the vaccine is going to be better. Okay, I got it.
Because I'm like, okay.
That's what I was going to get to.
You were
I'm getting the
building up
details.
and building up. I just couldn't take it anymore. I'm sorry.
Okay, no more building up.
Which one?
It's not that the naturally acquired immunity is not good. It's that since the vaccine is so specifically or vaccines, I should say, are so specifically targeted to the spike protein, it gives the potential for antibodies to be better able to respond to variants that even have single point mutations in that binding domain, which is in the spike protein.
The real reason this is an important issue is because more people, at least around these parts, where you're sitting, I think, have got the vaccine than have recovered from. So, if both groups are feeling immunized by this, and one really isn't, that's an important thing to know.
Yeah.
But it also, we still got to keep an eye on this Delta variant thing, though, because it seems to be finding.
Yeah, and the Delta variant, it's definitely a variant of concern. It is massively more infectious. Reports in Australia and other places are laying it out to be, instead of having to be in the space of the virus for 15 minutes to be able to get infected, they're saying you can be infected within a minute, within a few seconds of being in contact. Just walking past somebody and breathing their air for the Delta variant could be. But that said, vaccines are very effective against the Delta variant. So at this point, it's the unvaccinated population that is really the population of concern. And that's kids, and that's people with immunodeficiencies. It's, yeah.
Well, and there's still people who are just ignoring it, despite the constant coverage of the COVID pandemic everywhere you look, all over cable, network, radio, news stations, every website, portal, platform, and social media site. Every
conversation that you have with anybody.
Well, every conceivable way in which information is disseminated in this modern era. But there are some people who failed more than others to take these necessary precautions. New research from West Virginia University sociologists looked into excess deaths among the Amish population in 2020. Oh. Did anybody tell the Amish? We had it on all the electronic devices. They don't have, maybe they just didn't hear that this was happening. What's that? Oh, that's what the Mennonites do. Okay, I didn't know. So it turns out Mennonites are basically Amish who are basically not Luddites. They can have electronic devices. Yeah, so the death rate during the first year of the pandemic soared above baseline average. The largest spike being last November. They had 125% higher mortality than normal. Research led by Rachel Stein, she's an associate professor of sociology, analyzed obituary information published in Amish Mennonite newspapers to examine excess deaths among this segment of our population in 2020. The results are published in the Journal of Religion and Health and explains co-author Katie Corcoran, associate professor of sociology at West Virginia, by taking multiple years of historical data, we can create an average rate of death. For 2020, we identified how much extra death occurred on top of that average, which we call then the excess death. And then Stein, who's leading the study, grew up in Amish country, decided to explore COVID-19's effect on that community, pointing out that, the community members really weren't practicing social distancing at the outset. Amish community do not use modern technology, such as electricity even, and they tend to distrust modern preventative medicine. Stein said, there's a perception that COVID is like the flu there. If people get sick, then people get sick, and we'll eventually get over it. Co-author Corey Collier, associate professor of sociology, views the team's findings through the intersection of religion and government guidelines, stating that mandates prohibiting church can backfire. When this pandemic hit and the state prohibited worship, well, they prohibited gatherings of people, but that affected worship, they didn't take it lightly. They don't look at it as nanny state or as a government overstepping bounds. They take it as persecution. Any attempt to curtail religious gathering and practice interferes with their sacred business. When states step in and say, you can't gather for church, they say, well, there's man's law and there's God's law. We're going to follow God's law. A study suggests that in general, many groups complied with government guidelines, limiting religious gatherings in March and April, but many then resumed church services by summer. And this is just my observation. It seems to actually show the effectiveness of these government guidelines, because the study's pointing out that when the government restrictions became more relaxed in the fall, that's then November, that's when researchers observed the greatest spike in the excess deaths. So there must have been a decent amount of awareness in the community to at least have done the moderate reduction of group sizes. So he says they think the future is going to remain problematic in these communities. Most members will not practice the social distancing. They're not getting vaccinated, the research team said. They said, though, that the Amish are a great example, but they shouldn't just be singled out because the methodology of this study could be used for all sorts of other religious groups. There's
religious groups, and also just as a reminder of the fact that not everybody is connected to everything all the time. There are lots of people who are slipping through the cracks. And it's not just communications cracks. There are people who are in, grocery store deserts, right? Where there's just there's no pharmacy anywhere near them where they can get a vaccine. So it's like, how much is available? How much, first is the information available, and then is the opportunity available? There's a lot of these things that kind of tie in there.
Yeah, that's true. But I didn't know that about the Mennonites. That's I learned something about the Mennonites. It does explain why the Amish girl was excommunicated. To Mennonite.
Oh, Justin.
Nobody's going to send me an evil email about it. I won't get any negative tweets over that one.
Because it's going to be cut out of the podcast.
Because it's going to be cut out of the podcast. And because, very likely, Mennonite probably wrote that joke. The Amish aren't going to hear it. Yeah. Mennonites, they have a wonderful sense of humor.
And okay, but on this whole conversation of opportunity and who's seeing these messages and who's hearing about COVID-19, there's also the question of hesitancy. Some researchers towards even getting the vaccine, and some researchers just published a study which is a review of a whole lot of surveys. It's a systematic review and meta-analysis of acceptability and its predictors. Surprisingly, out of 38 articles with 81,173 individuals included, the pooled COVID vaccine acceptance rate was 73.31%. Studies using representative samples reported a rate of 73.16%. The pooled acceptance rate among the general population, 81.65%. It's really high. However, this was higher than that among healthcare workers, which was 65.65%. What is going on in healthcare that healthcare workers have a lower vaccine acceptance rate than the general population, according to these surveys? I just would love to know the nuts and bolts of the surveys, and this is fascinating to me. It maybe explains why several hospitals are having to convince their healthcare workers to get vaccinated, why several healthcare workers have chosen to leave their jobs because they didn't want to get vaccinated for COVID-19. But anyway, people who received an influenza vaccine in the last year were more likely to accept COVID-19 vaccination, and national and individual level interventions can be improved to get better vaccine acceptance, understanding that the main reasons for willingness are protecting oneself or others, and the main reasons for hesitancy had to do with side effects and safety. So there are very specific messaging efforts that can take place as a result of this data, which is pretty exciting. And I'm
oddly not surprised, though. The healthcare is education it is interesting. It is, but it's, I think, because healthcare, nursing education, this sort of thing is not approached like you were learning science. It's approached from a very vocational, manufacturing-esque trade. And it's less on the science behind why you're doing things and much more focused on process and procedure of performing a functional duty to the manufacturing of hospitals. So I'm not actually that surprised that just by being adjacent to medicine does not.
Yeah. I don't know. I find it very interesting. I think that it is obviously something that needs to be addressed very significantly. And then finally today, also the CDC did report they had a panel look into the question of whether or not myocarditis is being caused by COVID-19 vaccines, and they have said that there is very probably, there's a very likely link, although it's a very rare side effect. But it is again similar to the Johnson & Johnson AstraZeneca blood clot side effect that was seemed to be very age and gender related. This also appears to be more age and gender related. However, the myocarditis is in the younger age group and mostly boys. And at this time, they have certified about 250 cases that they think are linked to the vaccine, but no deaths at this point. And then they are continuing to look into other cases that have been reported to the adverse events reporting system. So it's all a process, but it's a very rare symptom and mostly treatable and fine. But something to think about. This is This Week in Science. We are a science podcast, and we thank you for joining us for this episode. Thank you for spending this time with us. If you appreciate the podcast, please consider heading over to twis.org and clicking on our Patreon link. Become a patron of TWIS. Support us and allow us to do this wonderful broadcast on a weekly basis. We really appreciate your support and can't do this without you. We're going to come on back right now to Justin's Animal Corner. It just feels so wrong,
with
Justin.
No, you just got to put your heart into it. Just try it. Justin's Animal Corner with Justin. Doesn't like bats.
Justin, tell me some animal stories, Justin. Okay.
Oh, Blair, we miss you. We can't do
so
this without
much.
You. But since you're gone, best known for its presence in house cats and a tendency to infect and alter the behaviors of rodents and humans, its legacy as being the culprit behind most newborn blindness, the parasite Toxoplasmosis, which only recreates in cats, domestic cats mostly, is now also being associated with bold, deadly behavior among wild hyena cubs. This wild behavior puts them at greater risk of being killed by lions. Finds new research from the University of Colorado Boulder.
My goodness. So, wait, Toxoplasma, hyena cubs, eaten by the lions, and then it passes through a cat species.
Yes. This is why the cat lady has cats. It gets into a mouse, it makes them less afraid of cats. It gets into a rat and makes them less afraid of cats. It gets into a hyena, the mortal enemy nemesis of a lion, and it makes them fearless of lions. Why? Because parasite's got to complete its business regardless of who the host is. So yeah, this is they published this week in Nature Communications. Reinforces previous research which has found the parasite prompting these sort of profound behavioral changes in hosts, and is currently around 2 billion people worldwide are infected with it. T. gondii has been well studied in laboratory settings, but this is one of the first times they've been able to do observations in the wild. This is Quotey Voice. Let's see, who have I got here? This is Zach Laubach, who's a co-lead author of the study, postdoctoral fellow in ecology and evolutionary biology. This project is one of a handful of long-term continuous studies on a long-lived mammal. Our findings suggest that infection early in life leads to bolder behavior and is particularly costly for young hyenas. So this also happens to be the one place in a hyena's life when they can easily take down a hyena. The older hyenas, interestingly in this study, learn to be more cautious about lions. But in a group, a bunch of hyenas can be a real problem for a lion. So it's can be a fair fight on some level. But when they're young hyenas, they don't have a shot under a year old. Multiple strains of T. gondii found throughout the world infecting warm-blooded animals, including humans who have house cats. That's a big spike there. So during different life stages, through contaminated soil, drinking water, eating meat of other animals who've been infected, it can be passed down even from mother to baby, which we've talked about in the past, is I think the leading cause of childhood blindness. Anyway, for infected cubs, hyenas, up to 1 year old, they found infected animals are bolder, approaching lions from closer distances than the unaffected cubs. And that infection among cubs also corresponds to the higher probability of being eaten by lions. In the study, lions were responsible for all the hyena cub deaths among their infected animals. 17% of unaffected cubs died before the age of 1 due to lion attacks. All hyena cub deaths among infected animals. Yikes. But after they're 1 year old, no difference in how close those lions got, regardless of their infection status. Some scientists theorize that this parasite manipulates its host behavior, this is what we're talking about, in order to get back into cats, where it can sexually reproduce. But the data from this study, they're saying, does not provide enough evidence to disentangle the theory supporting the adaptive mechanism for the parasite from other plausible alternative theories, although it is another one. It seems pretty
straightforward, but like we've seen many times in science, what seems straightforward is not always. So
I was convinced by the cat lady who's got the 50 cats in the house. It's just the parasite's just like, get more cats, bring more cats into the house. You need more cats because I'm going to need that many cats to take down a human.
The Toxo needs all the cats to take the humans down.
There's a species of butterfly found in sub-Saharan Africa that is able to migrate thousands of miles to Europe. It crosses the Saharan desert when the weather conditions are just right. And it's set the world record for the furthest migrating insect. The striking painted lady, Vanessa Cardui butterfly, has been shown for the first time to be capable of making the 12 to 14 million meter round trip, the longest insect migration ever seen. In greater numbers, they can do this when wetter conditions in the desert help the plants on which they then lay eggs. And yes, for those not familiar with meters, 12 to 14 million meters is about 12 to 14,000 kilometers. It's pretty much the same. Professor Tom Oliver, ecologist, University of Reading, and co-author of the study, said, "We know that the number of painted lady butterflies in Europe varies wildly, sometimes with 100 times more from one year to the next. However, the conditions that caused this were unknown. And the suggestion that butterflies could cross the Sahara Desert and oceans to reach Europe was not proven. This research shows this unlikely journey is possible and that certain climate conditions leading up to migration season have a big influence on the numbers that can make it. It demonstrates how the wildlife we see in the UK can transcend national boundaries, and protecting such species requires strong international cooperation." So, yeah, the study is published in the Proceedings of the National Academy of Sciences journal. The team calculated that butterflies must fly non-stop during the day and rest during night to cross the Sahara, making maybe a few stops to find a little bit of nectar to feed on. This actually is a similar pattern they have seen before in songbirds that migrate. They also conclude that the butterflies must fly up to 1 to 3 kilometers above sea level to take advantage of favorable tailwinds, because if they're just relying on their own flying power, they might not be able to make it that distance. They calculated the painted ladies have enough body fat after metamorphosis to sustain 40 hours of non-stop flying.
Wow, that's amazing.
Yeah.
It's very similar to the research that we heard about from a previous guest talking about mosquitoes and the flights that mosquitoes make, where mosquitoes get lofted up by winds and they make these 1,000-mile trips. And so there's this definite possibility that mosquitoes transit the Sahara Desert, lofted on the winds over thousands of miles. And this is happening with the painted ladies too. It's just it's amazing. Thousands of kilometers.
So, but one of the other sub threads of this story is that they do need a certain amount of the right weather conditions in the Sahara.
Yes, to be able to
for the trip to be possible. And if it doesn't happen, they don't go, or not as many of them endeavor upon that journey.
I wonder how much of it is I'm a butterfly who migrates and I'm going to try to migrate versus the winds are favorable. Maybe they will take me, and have it being like more of a chance event that happens around that time of the year because the conditions are more favorable. Yeah. It's
an interesting story to me too, because I have a family connection to it. My ancestors were lumberjacks in the Sahara forest.
They caused that desert? What?
They were good at their job.
No.
Is my last
right.
animal story? Research is being presented at the European Congress of Clinical Microbiology and Infectious Diseases. That's going to be held this year in early July, I think the 9th to the 12th, suggesting that 1 in 10 veterinary workers in the Netherlands carry strains of multi-drug antibacterial resistant bacteria, compared to only around 1 in 20 in the general population. Higher prevalence could not be explained by known risk factors such as antibiotic use, recent travel. It seems highly likely that occupational contact with animals in the animal healthcare setting may result in shedding and transmission of multi-drug resistant pathogens from animals to humans. Interestingly, for the study, though, they hadn't tested any of the animals. This was just a study of the healthcare workers. They didn't have a study conducted on the pets that they then were interacting with. Stool samples were collected from 482 veterinary workers. These were vets, technicians, assistants to the vets. Genetic sequencing was used to identify species of bacteria in the samples. The presence of extended spectrum beta lactamase and AMP-C drug-resistant genes were found. These are enzymes that don't get broken down, I guess, that by our drugs that we use to fight the bacteria. Anyway, veterinary staff completed questionnaires about contact with animals, this sort of thing. What's also interesting in here, so the veterinary workers were twice as likely than the regular population, the non-veterinarian population at large. But 17%, which is then almost double the veterinarian workers, of their household members also were carrying strains of the drug-resistant bacteria. Which is... It takes a minute to sort of get your head around, like, okay, so they're the veterinarian workers are having the close contact. Perhaps there's animal interaction. We don't actually know because they didn't look at the animals. And they're twice the general public's rate of infection with this drug-resistant bacteria. But their family members are almost twice the load that they have.
Right.
So what's the difference? The difference is the veterinarian workers are taking constant precautions at work, wearing gloves, changing gloves, washing hands, using alcohol.
Yeah. Right, cleaning their veterinary clothes even. Mm-hmm. Yeah.
Not enough to prevent getting twice, but enough to keep their conditions,
their numbers down,
suppressed.
Yeah.
Whereas their family members who might
Aren't.
stop by or might have interacted or might have caught it on the day when they didn't stop it from getting through
Or it could
aren't
be
doing those things on a daily
it's
basis.
all in the car that the family shares, or it's, you come home and you don't take a shower and it gets in the bed and then you're sharing it. It would get
it was a very
concentrated.
It was a smaller sample size
Interesting.
of the extended family members, so that's not part of the data that they're highlighting, because I think it was I'll get the real number. It was only 4 out of 23 people carried these bacteria, so that's
still really low.
It's twice the rate of the number of household members.
Yeah.
It's twice the rate of almost of the veterinarian workers, which then makes it about 4 times the rate. So it's significant enough to sort of.
It's a lot.
Yeah.
It's insignificant yet significant. And what kind of influence is this happening? Take more care.
Yeah.
is
No
the
samples were
bottom
taken
line.
from animals attending the clinics, so they couldn't connect the dot. But this obviously is a good place to do some future research.
Totally. Yeah, I hope they do. That's this kind of research is really important because multi-drug resistant antibiotic resistance is a huge problem, and until we can figure out how to deal with it and what we're going to be able to do, we have to figure out where that resistance is coming from. Where is it spreading? Where are the pockets? Yeah.
And that does it for Justin's Animal Corner. Remember to get your pets spayed and neutered. Actually, I guess you can't do that. Can you get spayed and neutered? I think it's
Not
one or
in
the
the
other.
same animal unless it's a hermaphrodite.
Okay, spayed or neutered.
Or
Don't
neutered.
Try to do both. And don't do it at home. Don't do it at home. You still need to go to a veterinarian.
Go to your
but when you
veterinarian.
go,
Yeah.
wear plenty of PPE. Gloves, mask. Don't just walk in there shaking hands, kissing your veterinarian.
Don't lick
not
your
safe.
veterinarian, folks. Alrighty, I've got some brainy stories for you. So, brains! I want to talk first about memory. Let's talk about how we remember things, right? The hippocampus is the part of the brain that's involved in incorporating new memories in. It's part of this channeling experiences that you have, events that you have, and taking that information and storing it away. Putting it away, making associative networks throughout your brain related to all the contextual information of certain events that happen. And in that, the hippocampus is a huge part of how we remember the world, where that information is. And the hippocampus does become active when we are remembering stuff, as it is part of that network of it's the librarian that told everything where to go, so to speak. Researchers have known for a long time that this is an essential part of the brain for memory, but a new study out of Northwestern University has determined that the hippocampus is also essential for constructing your present. The hippocampus is active all the time. And it's helping you to take in information, but it's not just doing the memory storage. It is this discovery is that the hippocampus is involved in comparing your present experiences against your past experiences to help you predict what might happen in the future. So the hippocampus is this essential organ in our brain to reality construction. It is basically going, oh, you know light bulbs. Oh, you know stairs. You know people. Oh, you don't know that. What's red? What's that? Danger, that's different. What's that thing? Knife in hand, is that danger? You know, and suddenly you have a new situation that is an event that your hippocampus is encoding, but it is helping you understand the present moment, which I think is a very interesting new function for the hippocampus, not just memory storage librarian, but also it helps you guide your decision making. It helps you construct your reality.
So that's very interesting. I was reading the study that about dreams when it described dreaming as, 50% reports a memory source were connected, or multiple past experiences were connected to a dream. They also found that 25.7% of dreams were related to specific impending events. So it was sort of trying to predict the future. 37.4% of dreams with a future event source were additionally related to one or more specific memories of past experiences. So it's a sort of interesting thing that your brain is trying to do. It's like, here's all the things that happened in the past. Here's the things that I know are coming. And let me run some modeling. Let me do some predictive modeling.
Right. So your brain is doing that predictive modeling while you're sleeping, but then it's also doing it in the current moment. So there is the whole, like, we are constantly, a quarter of a second behind what's actually happening in the world because of the fact that our brain is taking in information, and we are a quarter of a second consciously aware. Our conscious awareness of what's going on is lagging behind the construction of our reality by our brain. So information is coming in. The hippocampus is going, does this match with what we know? No, it doesn't match with what we know. And suddenly you're conscious of going, hey, I don't know what's going on here. Or you can drive a car and just kind of get lost in that dreaminess of driving because you've done it so many times before, until that moment when somebody runs a red light, right? Your brain is helping you constantly. It's a prediction engine, and it's all of these parts of the brain working together while you're sleeping, while you're awake, to predict everything, which is pretty amazing.
So, but that quick question. So is it still the hippocampus? Do you think that's going to be operating the dream study? When they were looking at this, they weren't doing any sort of that sort of a dive into what part of the brain was functioning. This was just people self-reporting things. I thought it was interesting because then you have that one where you've predicted a model of the future. Apparently, every night you're doing this all night long. And you're like, oh, deja vu. Oh, I had a dream where this happened. Well, yeah, but you also modeled all the things that didn't happen.
Yeah.
But is it, because we're talking about when we're asleep, but do you think maybe our hippocampus is still doing the same job all night long?
Yeah, it's going to be involved in that. So it's part of the system of storage. And so as your brain is reactivating and running through events, probably, we talked a couple weeks ago about how the brain runs through things kind of at multiple speeds faster than they actually happened and plays through them. And then it's in your dreams. It's not just playing through them super fast while you're not paying attention. In your dreams, it's like constructing new situations and putting new things together and playing with new possibilities. And so your hippocampus is probably part of that whole wiring system of how a
bit of a lucid dreamer. Usually remember a good part of the dreams, so I know that stuff's going on. But then it starts to bring up the question, like, okay, so what exactly is getting put to sleep? It's sort of like the id. It's sort of like the one that wants to choose what's doing. It's this conscious, what I call a conscious me. All this other stuff's still going on. It's just me that's getting shut off? That's not fair.
Yes. Just you. You, go away now.
Okay, shh.
We're speaking.
Okay, brain's in charge. You're on timeout.
Yeah, exactly. The brain has put you into a timeout. That's right.
So you're going to sleep too? No. We have work to do, child. Go to sleep.
We love talking about aging on this show and our constant efforts, or Blair's efforts, and I'm sorry she's not here for this story, Blair's constant efforts to fight aging. Well, a new study's out this week in the Public Library of Science by researchers at PLOS Biology, and these researchers, Kiang et al., they have discovered a link between a protein that's in red blood cells that allows red blood cells to deliver their oxygen to various tissues. It is a protein that's called adenosine receptor A2B, or as the acronym is so cutely spelled, Adora2B. I wish it were Adorab. too, but it's Adora 2B. And Adora 2B, they don't know yet whether or not it decreases in its levels as people age. But in this study that they did in mice, they discovered that if they had mice with Adora 2B or without Adora 2B, those without Adora 2B, as they aged, they had more cognitive decline, deteriorating memory, they had hearing deficits, which was very interesting, and they had larger inflammatory responses in their brain. So if they did not have Adora 2B, all these things got worse faster.
Because
why?
Apparently, the Adora 2B kept the retroviruses busy talking. Hey, yeah, just think you're adorable. Just whispering to the Adora. Didn't have time to send out inflammatory orders to the rest of the part of the brain.
Well, the Adora 2B kept delivering oxygen to the brain and letting the brain do the work that it needed to do. The metabolism was able to keep going, as opposed to having hypoxic conditions without oxygen or without enough oxygen, which would then definitely lead to panic, inflammatory responses, and potentially cognitive decline as a result of that. So this is the big question now, whether or not in natural aging, whether Adora 2B in individuals decreases with age. We don't know that for sure, but based on their studies and also other work in which they adjusted hypoxic conditions for these mice and determined that those with Adora 2B did much better than those without Adora 2B. It's obviously a potential target, and it's obviously something that is a protein of interest moving forward for the treatment of that terrible transition from youth to age.
Find it graceful.
I'm trying to be graceful about it. And finally, my final story I really wanted to bring just because of one particular aspect of the study. Again, it's a mouse study, and I keep thinking about your mouse story from last week about how all these studies, if nobody says they're in mice.
Don't put mouse in the title. People just run with it like it's proven on humans.
Yeah, so I'm really trying my best to make sure I say this study is in mice. Researchers at the Riken Center for Brain Science and the Riken Bioresource Research Center in Japan with State University of New York at Buffalo collaborators have created a model system in mice to study naturally occurring melatonin. They've published this in the Journal of Pineal Research. And they've found in this, what their main findings is that the mice with melatonin showed that natural melatonin is linked to a pre-hibernation state that allows mice to slow down their metabolism and survive when food is scarce or temperatures are cold.
Interesting.
Which is interesting. This is not the most interesting aspect of this study to me. The most interesting implication of this study is the discovery or the just overall awareness that lab mice do not have melatonin. Wild mice do have melatonin. They discovered as they were modifying their mice and trying to get mice with melatonin, mice without melatonin reproduce better. And so laboratories have been creating a selection effect for animals that don't have melatonin and who are used in studies of sleep and metabolism very often. We have created these populations because they reproduced better. So more babies, we're going to use these ones. You pick the ones that have the more babies to have the more babies, and then you have the lab populations without melatonin, which I just found is absolutely fascinating.
Yeah, especially because while you're telling this, I remember this study on melatonin in mice and how blue light or something affected like the mice's melatonin, and that led to cancer rate or something. Like, I don't know, a decade ago.
Right.
But now I have to question whether or not you should trust mouse studies on mice about mice.
At least when dealing with melatonin.
Yeah.
Are they dealing with the right population of mice? Because obviously mice are lab animals that have lots of genetic strains that we have picked and prodded through for many generations, mouse generations. But
if
this
an
is
accidental selection process in lab mice, then that could be a problem across a whole bunch of
things. And I hadn't realized this, and I found it very interesting. But anyway, the evolutionary advantage to having melatonin, it protects wild mice from losing weight. They have this pre-hibernation torpor when they can't find enough food. So if they're in, it's wintertime or it's cold, they can't find enough food, they can survive and they don't lose weight because of this torpor. But lab mice, they don't have melatonin. They don't need it because they're in these wonderful environments that are temperature controlled, and they have ad lib food usually. So they're nice and chunky and very healthy, and they don't go into torpor. So lab mice and these wild mice with melatonin have very different metabolisms. This is really important for mouse science. But
Arnalor in the chat room is very succinctly pointing out, how often have we had stories about stress in lab mice?
Very often, yes. So I wonder who will pick up on the melatonin, this aspect of melatonin in lab mice, if it will be picked up on. I think it is a very interesting study for many reasons, but they are creating a population of mice that have melatonin that can be studied in this model system for how melatonin interacts and creates behaviors for mice innately. Yay, who knew we had to put melatonin back in mice? But that was my last story. Brings me to the end of the show. Have we done it? Are we done?
I think so. I think we tight 90 minutes. That's pretty good. Once we're done editing out all of my things that I said, we'll be right at 90.
1, 2, 3. We're there. Anyway, if you have any questions for us, you can always let us know what questions. If you have comments, send us an email and we can read it on the show also. Really appreciate the fact that you were here with us for the show. I do hope that you enjoyed it. Shout outs to people who help the show. Fada, thank you for all your help on social media and with the show notes. Identity4, thank you for recording the show. Gord, thank you for chat room help. And I also want to thank Rachel for her amazing assistance in helping me with script supervision. She helps get the show made. I also want to thank our Patreon sponsors for all of their wonderful support of this show. We really can't do it without any of them. Thank you to Pierre Velazarbe, Ralph E. Figueroa, John Ratnaswamy, Kira Carl Kornfeld, Melanie Stegman, De Kramsta, Karen Tazi Wooden-Yemes, Andrea Bassett, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Schneider, Donathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Shubru, Donald Mundus, Stephen Albaran, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredis 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack Bryan Carrington, Matt Bates, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard Pearson, Richard Brendan Minish, Johnny Gridley, Kevin Railsback Flying Out, Christopher Dreyer, Mark Massaros, Artyom, Greg Briggs, John Atwood. This profile name is hilarious in the context of some other podcast. I wonder which one. Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul Matt Sutter, Philip Shane, Kurt Larson, Mountain Sloth, Jim Drapeau, Shara Chavez, Stu Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Pecoraro, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you. for all of your support on Patreon. And if you would like to help out on Patreon, head over to twist.org and click on that Patreon button. Become a patron on next week's show.
We will be back Wednesday, 8 p.m. Pacific time, broadcasting live from our YouTube and Facebook channels, as well as from our website, twist.org/live.
Want to listen to us as a podcast? Just search for This Week in Science wherever podcasts are found. And if you... yes. And if you enjoyed this show, get your friends to subscribe too.
For more information on anything you've heard here today, show notes and links to stories will be available on the website, www.twist.org, and you can sign up for our newsletter.
You can also contact us directly by email. Kirsten at thisweekinscience.com. Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just put TWIS in the subject line. Stick it in the subject line.
Harder than it looks. I know.
Stick it in the subject line.
Get it in there. It'll work.
Yeah, stick it in the subject line so your email doesn't get, eaten by a butterfly bat.
You can also hit us up on the Twitter, where we are at Twist Science, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you'd like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
And have you learned anything from the show? Wait, no. I'm saying the different... I don't read this often enough. We will be back here next week. We hope you'll join us again for more great science news.
And if you've learned anything about the ending of this show, remember...
It's all in your head. Bum, bum, bum.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of Toxoplasma gondii. 'Cause it's This Week in Science.
This Week in Science.
This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science.
This Week in Science.
This Week in Science. This Week in Science. This Week in Science. This Week in Science. This Week in Science. This Week in Science.
Thank you all for watching. I hope that we hit all of your science hotspots this week. We got a little space, we got a lot of animals, got some Toxoplasma gondii, got brains, we got COVID. I think we hit a lot of topics in there. So thank you. Thank you for joining us. I think Justin's coming back. He gave me the 1 second. My nose is itchy. I didn't take my allergy pills today. He gave me the 1 second look. Thank you, Identity 4. I'm going to put you up on the screen there. Did we disprove the null hypothesis? R and Lore? Did we? Yeah, Eric Knapp, Blair is still sick. We don't know what's happening. She said she's going to go to the doctor tomorrow and have some tests. Yes, that's a week of really not feeling well. She said she started feeling better last week, Thursday, Friday, but then it came back with a vengeance, whatever it is. And yeah, so I mean, it's serious enough that she's talking to a doctor, so fingers crossed that they are able to figure it out, because if she's feeling so bad that she can't even TWIS. I mean, you gotta be feeling bad not to be able to TWIS, right? She's gotta be feeling really badly, and that makes me feel sadly. Yes. Yeah, I'm worried about her. I hope that she is fine. I'm sure it's going to be fine. Yes. Oh, yes. Adam Curry. Yeah. So, y'all, I am hoping that Night Attack invite me on to have the opposite conversation of the one that he had with Adam Curry this week. That I have so many thoughts. So many thoughts. Oh, the Podfather, he's like as bad as Joe Rogan. He actually listens to Joe Rogan. And he's quoting scientists who are known crackpots. Good night, Fada. Yeah, anyway, I like No Agenda, but yes, Shoe Brew, Adam and Dvorak are just, I love them, but they're, this whole COVID season, and they have lots more listeners than we do. They have a much bigger audience than we do, which really upsets me. Why can't I have a big audience to talk about the things that are fact-based for the science? I just don't understand. Why do the people with the crackpot theories who say, well, I researched a heck of a lot, and that makes me more of an expert than the experts, and so...
All right. So, but there is a very simple reason for it, and I've thought about this tremendously, because I think I kind of know what you're talking about.
You don't know what I'm talking about.
No, I do, because here's a chart that the researchers, you see this squiggly line? Well, they drilled a hole into the peak of Greenland, and because of this squiggly line, it means the climate change isn't real, because this stuff happens all the time. What I think science communicators, or scientists probably more so even than science communicators, what scientists get frustrated with, you being one of them, is the fact that these sort of crackpots get so much access. And the reason is smart people talk in a language that people don't understand a lot of the time. Grifters know how to present themselves as what people who aren't educated think a smart person talks like. They're very different.
No, I mean okay. So who I was talking about was a specific person who is not a grifter, and these are people who just think they know things, and they don't. They think they're smart because they work in media and tear media apart. I'm like, that's great. It's nice, but yeah, I do agree. It's the language use, and grifters for sure know how to use that language. And
grifters, but it's also the miscommunication that takes place when general media, general journalists even are endeavoring to ask questions, well-meaning though they may be at times, about science without having delved into the complexities of it. Like, for most of my part on this show, my role here is as a person. Like, I don't research
as a
guests.
person.
Good.
Yeah. I'm not a person. I'm a science robot.
I try to come here as
Dr.
a human.
Science robot.
I said person. I meant layman. I try to come in,
Yeah, I know.
with a level of education that doesn't presume the audience to know more than I knew before I researched the study and brought it, right? Before I read the thing and then tried to figure out what it meant. I try to bridge that gap as much as possible. But people, okay, here's the other thing. You look at the UFO conspiracy stuff, or the ancients, all these things where people are trying to take disparate pieces of information, put them together, and then somebody does in a way that they gravitate to. And maybe it has a story thread to it that makes it attractive. Maybe there's a little like conspiracy to it that sounds intriguing. But what I kind of gleaned from this is that people really want to know things. They really want to connect the dots. They have a hunger and a passion for knowledge and information. And unfortunately, many times the most palatable thing that they can access is, well, I wouldn't call it the stuff that comes out of the back end of a bull, because that actually has a use. That's fertilizer. That has tremendously positive uses that we can grow plants. And what they get is absolutely useless. And because it's accessible. So things like what we're doing here is an attempt to bridge those gaps, to make all of this information accessible. To talk about, I think one of the things that I appreciate about the show is that we cover so many stories that when even if you didn't care about a story or you did like one, or words that just by sheer numbers, a number of the stories start to sort of connect together. A lot of we have these threads of the microbiome. We have the threads of how your brain is processing information. We have these threads about all these things we were learning from the animal kingdom, and then they start to patch together, and then you begin to get a different view of the world that we're all experiencing together. Yeah. But if you went and looked up the YouTube person who's going to talk about, well, let me tell you what the real story is behind all these ancient civilizations, you're getting a bunch of useless information. And it's not going to help you perceive the reality in which you live. And it's sad. Yeah. It's sad.
It's sad. Yeah, I think what's it's upsetting that I'm typing something. It is upsetting that it's so easy for people to be swayed by a nugget of truth and a dynamic speaker, right?
Charisma. One of the is it Science Show? Who are the Green Brothers?
Oh yeah, Hank and John Green. Yeah. Yes.
Yeah. They put up a thing like it's become too hard to deny that blank has killed blank, and you're supposed to fill in the thing. And
fill in the blank.
Fill in the blank. And I posted that it's now impossible to deny that rhetoric has killed rhetorical conversation. That was my contribution. And like there isn't real debate or real conversation that takes place on most venues.
Yeah.
It's usually a feedback of people talking with people who are either like-minded or, oh gosh, like painfully open to the idea of whatever is being expressed. I mean, it is a way of hosting a show where somebody comes on and you just accept everything. You suspend all doubt and you accept everything that's being said. And you inquire about it as though it's just as real as anything else in the universe.
Yeah, I'm going to try and find the study that I found earlier today, but there was a study looking, I think, at the sentiment of people's social media posts, and basically people who have like more aggressive, negative, like combative social media posts against some kind of perceived opponent are more successful, more likely to go viral on social media, which I'm like, oh yeah, that makes sense.
Guava Sharma says it doesn't have to do with education level. Okay, so I disagree with you. Here's why.
Actually, with Adam Conover, yeah.
I think it does have to do with education level on the subject. That like, so you can know everything. You can be an engineer, okay, over here. You can very educated in this, but the thing you're likely to fall for grift on is something that has nothing to do with engineering. If they were talking nonsense about engineering, you wouldn't for a second be fooled by it. You would recognize this is nonsense. It's then when you're out of your field, whatever your education is, because education can be narrow bandwidths, okay, of education. It can be rather specific stuff that you've studied on. And I think we were talked about this almost when we were alluding to the healthcare workers not wanting, the percentage of healthcare workers who were adverse to getting vaccinated being rather high.
Yeah.
That makes perfect sense to me. Their bandwidth, if you look at what is taught in nursing and healthcare these days, it is not the science behind why they do their job. It is the function of the job. It's a trade at this point. So you leave. Yeah. But if somebody was coming up and saying, here's how you do an IV, and it's totally not right, they're going to see it immediately. They're going to be like, no, that's nonsense. I would never do that to any of my patients. But it's the
Here's
bandwidth.
how you cut a straw and make it sharp, and then you try and stick it in there.
No, I got an IV not too long ago, and the nurse explained, she really did know her stuff. She was explaining the valves within the veins that I didn't really know existed. I just pictured veins as these big long tubes, but there's all these pressure
series of tubes.
But it's a series of tubes with a series of valves, and sometimes they gotta punch through it. Yeah. Sometimes they gotta punch through it with the needle. They got a little resistance. Push it a little harder. Usually bust right through. Okay. Hopefully you don't leave the vein again. That's when you gotta Oh gosh, how'd it go? Well, no, you were fine. Okay, good.
I found the study I was looking for. It's in the Proceedings of National Academy of Sciences, and it's called Outgroup Animosity Drives Engagement on Social Media. And what you're talking about, the like the end of rhetoric or the end of rhetorical conversations because of rhetoric. What they say in the significant statement for their paper is that social media may be creating perverse incentives. And I like that combination of words, but perverse incentives for divisive content because this content is particularly likely to go viral. And they report evidence that posts about political opponents are substantially more likely to be shared on social media, and that this outgroup effect is much stronger than other established predictors of social media sharing, such as emotional language. These findings contribute to scholarly debates about the role of social media and political polarization, and hopefully can inform solutions for healthier social media landscapes. Language about outgroups are very strong predictors of angry reactions. Language about the in-groups are strong predictors of love reactions, reflecting in-group favoritism and out-group derogation. The out-group effect was not moderated by political orientation or social media platform, but stronger effects were found among political leaders than among news media accounts. In some, out-group language is the strongest predictor of social media engagement across all relevant predictors measured, suggesting that social media may be creating perverse incentives for content expressing outgroup animosity. Because we have found that that people respond when we're angry and we get mad with others, we're making those divisions even stronger. The rhetoric. Yeah. I don't know. Come on, people, stop being people. Can we just I really want people to rally around the whole joy germs thing. I've been saying this one for years. Hashtag joy germs.
I don't know what is joy germs.
Instead of sharing the mean oppositional content or combative content, share joyful content. Spread joy germs.
So, are you just saying say it with a smile? Or...
No. Find a way to say things positively instead of aggressively or negatively. If you do have something to say about an outgroup, does it have to be mean and attacking? Can we start trying to heal some of these boundaries?
Well, the thing is, I think because of it's a feedback loop kind of a thing.
Oh, Paul. Yeah. Paul Disney's asking, sometimes I wonder if human minds culture was ready for the internet. And the narrator says, no, they were not.
and then the subtangent narrator pops in and goes, well, it was invented by scientists for scientists. So those people were ready. Those people were ready. It wasn't meant for everyone.
It was meant to share the gene data bank data. It was meant to share all the physics data. It was, yeah. Oh, man.
Paul Disney says, if you can't say something nice, don't say anything at all. Oh, he's quoting my mom. He's quoting, or not my mom. He's quoting his mom. My mom said something very similar to your mom.
My grandmother said that all the time.
My mother said something very similar. She said, my mother said something very similar to me growing up. She said, if you can't say something nice, it means you're a Republican.
Justin.
This is how I was raised. I don't know how to change. That's the problem. It's hard.
I wonder how you ended up the way you are.
Well, I'm telling you, part of the problem though is, and I say this as a person who has only chosen to support one of the political parties because the other one is so bad, not out of unbridled love for what is there. But gosh, they believe in magic beans. They believe that somebody's going to come to the table and negotiate with them and hand them and the magic beans that they pay for are going to turn into something wonderful and it'll save the farm. But they're wrong. They're in a combat situation. They really are. It's sad, but that's what's happening. Another favorite quote of my mother's, of me quoting my mother, never suffer fools.
That's a great one.
I like that one.
It's like, all right, I gotta go now.
Yep.
It's like,
yeah. If you look to the left and the right and you don't have anyone there that you'd want to talk to, don't be there.
Yeah.
Okay. Good plan.
Am I in a place? Nope, don't need it. And this is the same for the internet. If you are scrolling through Twitter, Facebook, and you're looking around and you're like, what? Don't dive into the debates if they seem foolish and a bit crazy, right? Don't suffer fool. I want to say it like Mr. T would have said it. I ain't suffer no fools, right? Wouldn't he have said something like that? Mr. T was the best.
He said, "I pity the fool." Wasn't that?
Pity the fool, but yeah, because he would womp on them.
Because he had love for everyone. I even pity the fools. Yeah, you gotta have love for everyone. I think that was his message. I love everybody.
My grandmother,
and eat my cereal.
That
was his other
my
message.
Grandmother's other phrase was "Ikapila
Oh, don't pick your nose.
That's the one. Ikapila Yes. It was either that or you don't have anything nice to say, don't
Gallagher
say
got his first shot while wearing a TWIS mask.
Wait, who did? What? Where?
Yeah. Has the second one now. Gallagher is
Jerry Gallagher.
You are in Ireland.
Yes, he's
right?
in Ireland. Mm-hmm.
Got the first shot while wearing a TWIS mask. That's way to represent. See, and that's part of the in crowd, so we love him. And then if you didn't do that, then we don't like you. So where's your? I don't know. I'm still working on the in crowd, out crowd thing.
I want to like everybody, but then I meet fools. Like, oh, that's right. Yes, clowns and jokers. HNEK, that's right. Clowns to the left of me, jokers to the right.
Here I am.
Here I am, stuck in the middle with you. Do, 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 do. Stuck in the middle with you. Did I see Mr. T in Freaked? No. Did I see Freaked?
Is it weird to ask, is Mr. T still alive?
Yes.
I haven't checked in. I haven't checked in on him in a really long time.
Yeah. Last I checked, he's still alive. Yes.
Okay.
Gaurav, is that my quote? Did I say that? I think I did. Yeah, new twist. I would wear that. I would wear that shirt. I really would. It's true. Oh my goodness. Twist t-shirt. I want to like people, but then I meet fools. I don't know. This Week in Science. Don't be a fool. Don't be a fool. Oh, geez. Yeah, people with a poor immune system. Right. If you don't have a strong immune system, then you're not going to have as much of an immune response to the vaccine. You're not going to have as many antibodies, even if you do get vaccinated, and will therefore be less protected. So that's the thing. It's not about you. It's not about me. It's like having to think in a more social context. And I think that flies in the face of a lot of American values. which individuality and independence, freedom is a very important thing. It's why I think we have such a huge issue with vaccine, anti-vaccine populations. It's not just fear. It's not just concern about side effects. It's, don't tread on me. Don't tell me what to do. I can make my own decisions. Yeah. It's about society. If you can't do something for society, maybe you should go live in the forest away from society. We have to take care of the people who are in the society with us who aren't able to get vaccinated. I don't know why this is so hard, but I do know why it's so hard. It's like I can start to understand the mental space that people are in, but then at the same time I'm like, but come on. Let's come together and sing Kumbaya and everyone get vaccinated. We'll get over the pandemic and then we will fix climate change and we will stop all wars and everything's going to be great. And then we'll meet our alien friend neighbors. Oh, and maybe they're not friend neighbors. And then the world community will have to band together to be able to go into space.
They won't. We just had the best example of a thing that the world should have got. Like, here was our alien invasion came in the form of a virus. We should have been like, regardless of politics, regardless of spiritual belief, regardless of eco-socioeconomic status, language, whatever the things that divide us, let us together defeat this common enemy. And it created nothing but division.
Yeah.
You know why? Because humans. Tribal apes.
Tribal, we are.
It's really...
I wish we could get past the, minuscule tribal divisions and start,
So
human tribe. Human tribe,
yes, right. So,
common goals. Survival is humanity.
My now 14-year-old daughter has often, she's talking about Star Trek. And every time it's like Star Trek comes up, her first thing that she always talks about is like, yeah, isn't it weird that in Star Trek, every time they go to a planet, the people are like a country, but they're the whole planet. And I'm like, well, at first I'm trying to explain, back then, they're actually trying to articulate different political ideologies of countries in the form of other planets.
Yeah.
But then I realized, actually, this is part of that, what is it, the Fermi test or whatever it is, that determines whether or the paradox, that determines whether a planet becomes spacefaring. Right. Until you become like one country as a planet, you will never get off the ground. You will never agree enough to work together for a common humongous effort, like exploring the rest of the galaxy.
I think that's true. Yeah. I don't know. What was I thinking of? Oh, have you seen, speaking of all these divisions, have you heard about the Freedom Festival?
Nope. It's anything with the name festival in it, I'm like, I'm not going to be interested for a few years.
No, just look it up. Just look it up. I'm
not interested in a concert, not interested in a festival. I'm not even that into a comfortable get-together amongst acquaintances.
Yeah, you don't need to go. You don't need to go.
Although, the one thing I am regretting, I regretted it last year, and I'm regretting it this year, too. You know what I'm missing?
What?
Santa Fe Institute.
I am missing the Santa Fe Institute so much. That was really fun.
We had an invite while we were in Seattle at the outbreak of COVID, and they were like, "Wow, we're going to see you. Come back this summer. We can't wait to see you." And of course, that didn't happen, and it's not going to happen this year. I had such a blast with their venue. That's like the one thing, even in the midst of all of this, that I'd be like, yeah, I'll go. I won't see my family for a few weeks after, maybe. This is like pre-vaccination. Won't see my family for a few weeks after.
Oh, no, I
I'll
got
go
it wrong.
Quarantine.
Redneck Rave.
Oh.
That's the one you have to look up, not Freedom Festival.
I don't.
Redneck Rave.
I don't. The fact that butt rock, if you know what I'm talking. It's like when the radio stations go, we're the radio station that plays nothing but rock. And then... and then it's all of these like entitled like stilted adolescent angry boy
Angry.
Screecher, Speaker, Weezer, whatever butt rock bands that are like, yeah, they're getting into the mic with the
Come
same
on,
guitar
Weezer
riff.
is not a butt rock
Yes,
band.
they are. Maybe
No.
it's not Weezer. Okay, fine. You can have your
Weezer.
Weezer back. But so this industry of music that, by the way, when they looked at it 15 years ago and looked at it today, musically it had not evolved or changed. They're literally screaming over the same lyrics that they were 15 years ago, or the same music. But it actually often is outselling what is considered popular music.
I love rock music.
Oh, so does the audience. I will. If you listen to me talk long enough,
I will spend
offend
days listening to nothing but rock.
Oh my god. Yeah, well, you're from Stockton, so I guess that shouldn't
right.
be surprising. Stockton, home of butt rock in California. So yeah, if you listen to me long enough, I will find a way to offend something that you hold dear.
I think I think that's true. Usually, I don't know that you've ever actually offended me. I think I'm very hard to offend, like actually offend.
Okay, good.
Yeah, this is one of my traits. I'm like water. Like what? Why are people mad? Don't get mad. Just go, whatever. I don't mind. I think people are angry too much these days. There's a lot to be angry at. There's a lot to be angry at, I guess. We got a lot to change. Oh yeah, Shoe Brew just went to Red Rocks. to a super spreader event. But that's awesome. Shoe Brew was a groupie for a band. That's fantastic. My cat's my Toxoplasma cat is here, meowing and saying hi, meow, meow. Hi, Cappy. Meow, meow, meow. Yeah. She said, "What are you doing down here?"
Yeah, you got a talker there.
Mm-hmm. She said, "What are you doing down here?" I'm just gonna meow at you, and I'm gonna sit at your feet. I'm gonna purr.
Oh gosh. I think I'm even allergic to cats over the internet.
No, you are not.
Yeah.
At least you're not as sneezy as last week. Oh my goodness.
Was it just last week? Yes, it was.
Mister, I can't even do this story because I'm so allergic.
We didn't. I don't even think we told the audience. We talked about it off air.
We didn't tell the audience at all.
It was the last story I was doing last week. And my eyes were watering so badly that I stopped being able to read the text from the story. And so I was like, so anyway, there's a story about whatever this was. And, okay, let's wrap up the show now.
I had no idea what was going on. I had no clue, just that he was rushing through this story. Yeah, you're rushing through this story.
I'm going to say the things I remember about this story before I lost vision live on the air.
And then I'm like, well, what about this? And you're like, yeah, I'm sure that's in there somewhere.
Probably part of, they probably looked at some of that. I don't know. I can't see. I can't read anymore.
Oh my goodness, that was funny. Allergies are good. Oh, Lil Nas X. That's right. I'm glad Justin isn't a pilot also. Yeah.
There's no cats in the sky. There's no cats in the sky. It's fine. It's about as far away from cats as you can get most of the time.
There's no cats on planes.
No. They're not allowed on my plane.
I met a cat on a plane.
What?
A passenger had his cat and it was a lap cat. And the
What?
cat, yeah.
I have never. Then I'm bringing peanuts aboard to the next flight. I don't care. Now I just don't even care.
Yeah, there was a guy, he had his cat on a leash, and
Really?
the cat was just basically hanging out on
this a
his lap
domestic
the whole
flight?
flight. And yeah.
Was it some airline none of us have heard of that
No, I think it was Alaska Airlines.
Really?
It was probably Alaska or
you know
United.
What? Yeah, I bet you it was Alaska. Because I bet you Alaska, it's because they're flying rural people with hunting dogs all the time. That's how I picture it.
What?
I picture an Alaska Airlines flight in Alaska being
Maybe an emotional support animal, the Marks.
the row. Yeah. It's like you got one row is the family, and behind that you got the hunting dogs. They're strapped in.
Oh, jeez.
And I also picture it as having gun racks on the plane.
Cats on spaceships. I think we should have cats on spaceships for sure. Absolutely. I want all the animals on spaceships, especially flying snakes.
All right. So I think it's time. That we all say get well soon to Blair.
Get well soon, Blair.
Get well soon. Quit
Get well
being
soon.
ill or whatever it is. Sorry
Be
with you.
healthy.
Get rest and recover.
Yeah. Don't get caught up in National Catfish Day on Friday. Tau Day is on Monday. What can we do with Tau Day? Do we make more pies on Tau Day? An excuse to eat pies. And yeah, I think next Wednesday is the last Wednesday of the month. We'll have gone through the 6th month of the year. That'll be crazy.
But it is it
stop
though?
the
Because
stopwatch.
I think
Make it stop.
I think every point in the year that we are in is just as crazy.
Yeah.
As the
Everything keeps going.
point that you're.
Keeps going and going. I need to take a vacation, like for months.
You sound tired. You know what? I think you should just say goodnight, Kiki.
I think that's what I'm going to do. But I think you should say goodnight, Justin.
Good night, Justin. Wait, how's it go? Now I'm forgetting because Blair says.
Now I say good night, Kiki. Good night, everyone.
Good night, Dr. Kiki. Good night, minions.
Good night, everyone. Thank you for joining us for another episode of TWIS. I am going to add a channel to our Discord so that we can get not just story recommendations and interview recommendations for the show, but also movie suggestions for us putting together a TWIS movie night. Maybe we'll start doing a TWIS movie night that we can all watch together. So I'm going to make a channel where you can put your suggestions in the Discord. Once again, thank you all. Have a wonderful week. Stay safe. I want to, Gord and Jerry, I hope that your side effects, if you're having any from the vaccine, second dose, that you're all good in the next week or so, that everything protects you. Get vaccinated, everyone. Let's try and beat these variants of concern and move through the summer with ease. It would be wonderful. Yeah. Thank you very much. Have a great week, and we'll see you next Wednesday, and hopefully Blair will be back. Good night.